already so we can already with this immunocytochemistry we can already see where in the cell we see expression of specific proteins. But there is another way where we can study chromatin dissociation. And we have already seen this in the presentation of William. So who remembers how we can study chromatin association other than by immunocytochemical staining?
Chromatin immunoprecipitation.
Where is somebody talking about that?
Almost.
Okay. Yeah, it's a very good way of doing this, and I will come back to that later on, because then you specifically look at this part of the DNA, but a more general way of looking at chromatin association.
EMSA?
Sorry?
EMSA, maybe?
EMSA, yeah, but we've seen one in the, so previously Willem already showed the differential extraction method. So that's of course one way, but I'll come back to the chip later on. So one way to look at this is the differential extraction, and then you can look at protein expression in different cell compartments. So how does this work? So again, we grow cells, and we make a lysate of it. And for this, we now use a very mild buffer, and this will only lyse your cell membrane. So therefore, you will end up with your cytoplasma in solution, and your nuclei will still be, if you spin down, they will be included in the pellet. So if you then spin down, you will end up with the cytoplasm, and the pellet you can dissolve again in the second buffer, which is a more strong buffer, and this will lyse your nuclear membrane. And then you will end up in solution having your nucleoplasma, and you will still have your chromatin, which is not soluble. You can spin it down again. And if you then sonicate your, add it again, new buffer, and then sonicate the fraction where your chromatin, you will be able to dissolve your chromatin fraction. And this again you will analyze just like a normal Watson problem. So then one of the next questions is which kinase controls chromatin association. So actually we've done a similar experiment as we've done with staining, we now look at differential extraction. And I want you specifically to look at this part of the gel. So here we look again at BMI. So and in the first, so these are just serum scarred cells, we already see chromatin association, and if we add serum, we see that it's lost from the chromatin. And if we then add a PcG inhibitor, that you see that it's probably prevented. And also, but more stronger with the MEK inhibitor. And if you combine it, then there's a lot of polycomb containing on your chromatin. So now we have the hypothesis that ERK somehow maybe interacted with polycomb and then potentially phosphorylate or somehow modify the polycomb, and that this is also important in chromatin dissociation. So again, the next question is how can we test whether there is a physical interaction between ERK and polycomb? Middle section. Here's something that I can. If we want to look at protein-protein interaction, what would be method to use?
It will be break knife.
Yes, that could be this. But if you already have two, because with your two hybrids, you would actually want to screen for new interactors. But if you already have two proteins which you want to know if they interact, what would be then more easy?
Hormonal
Right. So then you can do full immunoprecipitation. So again, so we grow cells and we make a protein lysate of it, and subsequently you will end up with a mixture of all kinds of different proteins. And if I would now be interested in the blue protein, I will add an antibody which would recognize this blue protein, and if I subsequently will wash, and during the wash test I will lose all my proteins which are not bound to the antibody, and I will end up with the blue proteins and also with all the proteins which are attached to the blue ones. So in this case, the green and the yellow ones. So And those again you can analyze on the Western blot. So that's actually what we did. So we did an immunoprecipitation against BMI1, and then we came back to probe for ERK. So this is the input sample, so this is ERK, so we see that ERK is also increased with stimulation, and if we pull down on BMI1, in the case of the star cells, we don't see any band appearing, so we don't break along ERK if we pull on BMI1. However, if in the both stressed and stimulated cells, we see that there is a band appearing. And this was actually the control where we didn't add the antibody. There is no band appearing. So indeed, we find the physical interaction between ERK and polycomb. So P38 phosphorylated serine 28, next to K27, and ERK somehow physically interacts with polycomb and thereby has a role in polycomb dissociation. So now to come back to one of the suggestions which the guy behind Sarah mentioned, the Chromatin immunoprecipitation. This is a way to study specifically on one locus. And of course we would like to know what happens at the specific ATF3 locus, because that's the one we are studying. So how does this chromatin immunoprecipitation work? So we go to cells. We fix the cells in the plate. And subsequently we lyse the cells. We end up with a cell homogeneous. And what we then do is we shear the DNA into small pieces. And you can either use enzymes for that, but what we usually do is we use sonication. So we have with sound. So we have a probe sonicator, and these sounds actually will shear the DNA into small pieces, which are around 200 base pairs. And because we fix the cells, all the proteins are attached to the protein, there will be still bound to your chromatin in this step. And subsequently, again, you add antibody, which will recognize the proteins of interest, but in this case, you don't take along other proteins, but you take along pieces of DNA, which are bound. The co-immunization is for protein-protein interaction, and this one is for protein-DNA interaction. But the principle is actually similar. So at the end, you will end up with small pieces of DNA which are bound to your protein's interest, and those you will analyze with qPCR, similar as you will do for the real-time PCR. So only you have different sets of primers because now you mostly look at promoter regions. So now we look at what happens with PRC1 dissociation at the specific ATF3 locus. So we did a chip, we designed primers for the ATF3 locus, and then we looked at, did the chromatin immunoprecipitation for two polygon proteins, CDX8 and PcG1, and we also looked at the polygon epigenetic mark. So first of all, by looking at the CDX and PcG1, we see that indeed there is they are present on the ATF3 locus in serum starved cells. This is the control, the background. And we see that upon serum stimulation that you lose both CDX8 and PcG1 from this region, which corresponds to what we've seen with both the differential extraction and with the immunocytochemistry results. So we lose PRC1. And what we also were curious what would happen with the H3K27 metal mark itself, whether it would also be lost. So therefore we also took along this immunoprecipitation, this chip, and we saw that there is actually no change between the serum starved and the serum stimulated cells. So apparently you don't need to lose the mark in order to lose the polycomb complex. So only the polycomb complex is teased off, but the mark stays there in order to, if afterwards the signal is restored, polycomb can come back at the same space, the same location. This is just an proof that indeed we see again an induction of ATF3 with real-time PCR. So then we've already looked at the PRC1, we also looked at the mark, but we haven't looked at the serine 28 phosphorylation. And that's what we also want to do in this case. So what we did is we looked again at the So in this case, I put in there, we also looked at H3, but this is another polygon target gene, and we've seen that there is no change in H3 K27 triangulation, but the H3 serine 28 phosphorylation is indeed increased. However, if we look at a non-target gene of polygon, so and the non-target is indicated by because there is no polygon mark present, so there is no, this is designed as a non-target polygon. We also see the same thing happening. So currently, this serine 28 phosphorylation, it is necessary, yes, but it's not specific to this polygon target gene. So it also happens at other places. But if you remember, we also need ERK signaling, so we need multiple things to happen at the same place. So we need this serine 28, and we need the binding of ERK, but this serine 28 phosphorylation also takes place at other sites. So then what we haven't asked the question what would happen if we inhibit or stimulate MP3. That was also one of the suggestions which we got previously. So that's also one thing we looked at. So then we took APF3 as a model, and first of all we overexpressed MP3. So with the normal situation, so the white bars is the normal situation, so you again see induction of your gene of interest. And if we overexpress MP3, actually we see less induction of the APF3 levels. So and vice versa, if we inhibit the activity of MP3, we see that there is more induction of APF3. And if we also combine this with the, so previously we already see that the ERK and the P38 inhibited, and we've seen, I haven't showed you the results, but we've seen that it's actually that the inhibition of ERK and P38 overrules the inhibition of MK, and this was also because we think it's more upstream in the kinase. But these results actually suggested to us that not only ERK and P38 are upstream of MP3, but that MP3 might also feed back as a negative feedback to actually also inhibit the signaling which was induced by adding these mitogens. So in order to study this, we looked at what would happen with the phosphorylation phase of both ERK and P38 if we change, if we inhibit the expression of MP3. So this is what is in this slide, and I just want to focus on the little red boxes. So here we did a time series of serum simulations. So we looked at control cells, the cells where we inhibited MP3. So normally you see that upon serum induction, you see that ERK is phosphorylated. So this is one of these examples of these specific antibodies for a phosphoprotein. So ERK is phosphorylated, and in time the phosphorylation goes down. However, if we inhibit MK, we see that it goes up, but it stays high much longer than in the control situation. So currently, this MP3 is normally inhibiting ERK, and thereby, like, so you induce its signal, and this will be inhibited again. So the cell is so sophisticated that also the signaling will be again silenced when it stops. And this is also what we see with the upstream kinase of ERK, so the MAC. So also we see the control situation is phosphorylated, it goes down again, but if you inhibit MK, you see that it stays up all the time. And one of the questions was, how can this happen? So how can MK inhibit ERK and MAC? Because MK3 is actually a kinase as well. So we thought that maybe MP3 induces
the phosphorylation.
Induces a phosphatase
action.
Yeah, and that's what we looked for. So there's not a lot known about it, but DUSPs are this is short for dual phosphatases, and they're involved in dephosphorylating and therefore deactivating MAC and ERK. So what we've seen is, in this case, we looked at MP3 overexpression and MP3 inhibition. So we looked at several DUSPs, And we see with dust 2 and dust 5 that they are induced by pathogenic stimulation, which is one requirement. And we see that they are much more induced if we overexpress MP3, and they are much less induced when we inhibit MP3. And this is not the case for all of them, but it's specific for 2 and dust 5. So potentially what happens, we have mitogenic signaling, it activates MAC, ERG, P38, this will activate MP3 polygon dissociation, but also it will activate DUFs, which will deactivate upstream kinases, and therefore they will silence again the signaling pathway. So then there was one last question, is whether we can show this in vivo. And one really good model for this, as we've already seen, is to look at this in fruit fly. So, and to show you a little bit, to come back on this yeast to hybrid principle. So what we have to do in order to be able to specifically look in the fly is that you have flies which actually have can express your gene of interest, and this can be actually regulated. So this UAS sequence can be bound by Cal4. Only in this case we don't split it up into parts. We just have one binding, one transcription factor binding, and there is. If this Gal4 is binding, then you have expression of your gene of interest. However, if there is no Gal4 present, then your gene of interest is silent. And if we have two flies, one expressing the construct and one expressing the Gal4, and we cross those flies, we will end up with flies which express both construct and Gal4. And what is the nice thing about this is that you can specifically regulate in which part of your fly will express the Gal4. So you can, for example, only express it in the wing. So that will mean that only in the wing that this will happen. So that you only get expression specifically in the wing. That's what we did as the last thing for the paper. So we looked at the wings. So here you see there is actually a real picture of the wing. And you see there are two kinds of tissues. This intravenous tissue, which is here all the gray stuff. And the black lines here, these are the beads, which are inside the wings. Now what happens if we overexpress either P38 or ERG? We'll focus on ERG, it's the lower one. And the ERG is, in fruit flies, they tend to call proteins with a different name. So it's rolled in fruit flies. And if you overexpress this, you see that there are little extra veins appear, which are indicated here with the arrow. If indeed, so there is another question for you. If we would expect that MP3 inhibits ERG, what would then be the experiment to show in this model system that it's also the case in vivo? So MP3 inhibits ERG. We see if we overexpress ERG, there's more veins. So what would you then want to study? And what would you expect to find if that's indeed? Yeah. No. If it inhibits ERG, more ERG gives more veins. It's exactly the opposite. So ERG makes more veins, and if you take away the break of ERG, this will actually accelerate even more. So if you overexpress MP3, then you would expect to find less. Follow me? Yeah. Okay. So this is what we did. Actually we have to
not really
see the situation as depicted here. So with overexpression of ERG, we see that there is already more veins than in the normal situation. However, if we also take away MP3, that's even much more increased. We see that there is more extra veins appearing. So to summarize this, so we've seen that phospho-ERG and MP3 are part of the polygon complex, so they interact. Phospho-ERG and polygon. We see that ERG and regulates PRC1 association, but not serum 28 phosphorylation. And we've seen that P38 targets H3 serum 28 for phosphorylation, and that the description requires that polygon dissociate, but that there is no result given 7 to be removed. And finally, we showed that MP3 potentially induces expression of dust, which would inhibit the upstream signal guidance and therefore re-silencing this signal gap. So, and of course, I didn't do all this research by myself, so there the entire group participated, especially Hanneke, and also our collaborators in Paris, Emmanuel and Frédéric, who did a lot of the flight stuff. I've also been there for about 6 months to work on that. And we also had collaboration with Japan, Germany, and also with the binary informatics. And this is actually, this paper was recently accepted in Epigenetics and Genomics.
All right, so this is quite a lot to take in, I guess. Be reminded that this sort of thing, interaction between proteins and genes, is exactly what I will be examining on at the end of this block. I guess there will be a practice session at one point in time. Typically what I do is I give all the information that you need to answer the question. But you need to rake your own brains in order to answer it. Now, we will make the handouts for this course available shortly. I didn't want you to have the handouts before we gave the lectures, because otherwise there would be a lot of interaction. Are there any questions for Peggy at this point? Things that you really didn't understand or don't believe at all? Wow. Basically what Peggy has done is she's taken the story a bit further. I left off at where we had established that there is some sort of interaction between, functional interaction between polygon and MP3, and Peggy shows that MP3 has a regulatory role in expression of genes which are controlled by polygon. So that's cool. All right, well, if there's no extremely urgent questions at this point, no, there isn't, I guess. Then we move on to the journal club paper. If there's any problems that you came across that I think I might want to help you with, now would be the time to bring them up. Who did not read the paper yet? Okay, who read the paper? Why? Excellent. And no problems? No?